Welcome back, guys. Now in this video of general physiology, we will be discussing about the transport of substances across the cell membrane. Okay, so let's write transport of different substances across the cell membrane. Okay, so how different substances will be transported across the cell membrane. Okay, the substances are crossing across the cell membrane. Now, first of all, you should know what are the two different types of transport. Okay, the two different types of transport are, one is active transport and other is passive transport. Okay, active transport and passive transport. So what does it mean by active transport? Active transport means in the transport of a substance, it can be any substance, in the transport of that particular substance, energy is utilized. Okay, active transport means energy is utilized. Okay, energy is utilized, okay, in the form of ATP. ATP is involved in the transport of the substances. That is active transport. Now, you might be having a doubt. So, why the energy is required to transport a substance? Okay, because here the substance is transported from low concentration to high concentration. Okay, usually substances will automatically move according to the concentration gradient. We know that a substance will move from high concentration to low concentration just like that. It happens. But if you have to move a substance from low concentration to high concentration, you have to give the energy. Okay, you have to provide the energy in the transport of that substance. So that kind of transport is called as active transport. Okay, so here the substances are moving against concentration gradient. Okay, here the substance is moving against the concentration gradient. So that's why you have to give the energy. Okay, now what is passive transport? Passive transport, no energy is utilized. Okay, there is no need of any energy. Why there is no need of any energy, sir? Because here the substances are moving according to the concentration gradient from high concentration to low concentration. Okay, so a substance is moving from high concentration to low concentration. So that's why the substances are moving according to the concentration gradient. So that's why there is no need of energy. Okay, substance moving according to concentration gradient. Now, having said that, you know there are two types of transport where energy is involved, that is active transport. A for A, active transport may ATP is utilized. Passive transport, there is no need of ATP. Now, here active transport is of for the two types. Okay, active transport itself is for the two types. What are they, sir? Primary active transport, okay, primary active transport and secondary active transport. Okay, primary active transport and secondary active transport. These are the two types of active transport which we will discuss in a minute. And the passive transport, may any subtypes are there? Yes, here also two types are there. One is diffusion. Diffusion is a type of passive transport. Okay, diffusion and osmosis. See, from now onwards, if someone asks you that diffusion, does it require any energy? If a substance is being transported, a gas is being transported via simple diffusion, if I am saying that, is there any use of any energy? No need of any energy. So, diffusion ke liye, diffusion and osmosis, they don't require any energy. They are passive process. ATP is not required, energy is not required. The substances are moving from high concentration to low concentration. Okay. Having said that, now let me add a few more points. See here, diffusion. Okay, here the diffusion is of again two types. There are two types of diffusion. What are they? Diffusion includes simple diffusion and facilitated diffusion. Okay, simple diffusion and facilitated diffusion. We will discuss about them in a minute. Now, let's start with the primary active transport. Okay, let's begin with the primary active transport. Okay, so what exactly is this primary active transport? See, in primary active transport, yes, of course, energy is involved. But the key word which I want you to know is there is a direct use of energy. Direct use of energy, direct use of ATP in the transport of that particular substance. The substance is being transported and the transport of that particular substance, in that site itself ATP is getting broken down in the transport of substances. Okay. So, can you give us any examples, sir? If you ask me, yes. Examples, what are the examples of this primary active? One example is sodium, potassium, ATPase or sodium potassium pump. Second example, proton pump. You would have heard there is proton pump in the gastric mucosa which secretes acid. Okay, proton pump. Okay, so there is calcium ATPase. Okay, say if you are a really good student, by this time you should have identified something. So, how to identify primary active transport? So, any transport where you see this word ATPase or pump, ATPase. So, wherever you see this word ATPase or pump, definitely that is a primary active transport. Okay. For example, say, let me talk a few important points about this sodium potassium ATPase. Okay, let me talk about sodium potassium ATPase. What exactly it is doing? We all know, say, here is a cell. Okay, we all know from our basics that cells are rich in potassium and extracellularly there is more sodium out. More sodium is present in the extracellular fluid and in the intracellular fluid there is more potassium. The cells are bags of potassium. Okay. Now, here there is a transporter. Okay, here there is a transporter. Now, what is this doing? What is this transporter doing? So, the sodium potassium ATPase, okay, the thing which I am showing in the green color, this is the sodium potassium ATPase. What it does is, it throws 3 sodium first, okay, 3 sodium out of the cell. The 3 sodium is going out. Means already outside the cell there is more sodium. Okay, already outside the cell there is more sodium. Inside the cell there is less sodium when compared to outside. Okay, so the substance here is moving against the concentration gradient. Sodium is moving from low concentration to high concentration. Okay. Now, for this purpose, it requires ATP. Okay. So, sodium is moving against the concentration gradient. And in the same way, this will bring 2 potassium into the cell. 2 potassium into the cell. Outside we know there is less potassium outside the cell. Inside the cell there is more potassium. So even potassium is moving against the concentration gradient. Potassium is moving from out to in. Potassium is moving from low concentration to high concentration. So this process also requires ATP. First 3 sodium will go out in exchange for 2 potassium. 3 sodium is going out and 2 potassium is coming in. So antiport. Okay. So what I want you to know is here, say this sodium potassium ATPase. Coupling ratio. They will ask you in many exams, they have asked this question, the coupling ratio. Coupling ratio. What is the coupling ratio? The coupling ratio is 3 sodium is going out and 2 potassium is coming in. So the coupling ratio is 3:2. 3 sodium is going out, only 2 potassium is coming in. Substances are moving against the concentration gradient. So that's why ATP is involved in this process. So it's an example of primary active transport, direct use of energy. And how to identify it? Wherever you see the word ATPases or pumps, they are all examples of primary active transport. Okay. Now, apart from this, what I want you to know is, so the sodium potassium ATPase, of course, it's a protein. Okay, sodium potassium ATPase, it's a protein. It's a protein molecule which is present in the cell membrane. It's an example of which type of protein? Is it a carrier protein or is it a channel protein? Is it a channel which is allowing the substances passing through it, or is it a carrier protein? See, the sodium potassium pump is a carrier protein. Okay, it's a carrier protein. Now, why I am calling it as a carrier protein? Because carrier proteins will undergo conformational change, shape change while they are transporting the substances. Actually, the sodium potassium ATPase, it is made up of subunits, alpha subunit, beta subunit. This alpha subunit, okay, alpha subunit, it actually moves, it undergoes shape change. Okay, this protein is not static in nature. Okay, this protein, it will undergo shape change by using of energy. Okay, this protein will undergo conformational change which helps in the transport of substances. Okay, for better understanding, let me show you. Imagine here I am showing you the cell membrane. Okay, here there is an alpha subunit. Imagine this is the alpha subunit, and here there is a beta subunit. Actually, this is easy. Okay, representation, easy way to understand. Here there is alpha subunit, this is alpha, and here is beta, alpha beta. Now first what happens? See, to the alpha subunit, towards the intracellular side, sodium will come and bind. Okay, sodium is coming and binding to the alpha subunit. Which side? Intracellular side. This is intracellular fluid, here is extracellular fluid. Okay, inside the cell, outside the cell. Now, at the same side, even ATP is binding here. Okay, even ATP binds to alpha subunit. So, by breaking down this ATP, the alpha subunit, it will flip. The alpha subunit is now flipping. So, when the alpha subunit flips like this, what happens? The sodium will be moved out. Okay, the sodium will be transported out. The sodium will be transported out. Okay, now extracellularly sodium is released. Extracellularly potassium will come and bind. Again the alpha subunit, it will flip back to its original position. So potassium will be brought into the cell. So it's the alpha subunit which is kept on moving, flipping. So this sodium potassium ATPase, it undergoes conformational change. So that's why those proteins which undergo conformational change, shape change in the transport of substances, they are called as carrier proteins. So carrier proteins undergoes conformational change. Okay. Now, which subunit? Alpha subunit, it's doing the job mainly. So this is called as a functional subunit. Okay, alpha subunit is called as a functional subunit. Okay, so to the alpha subunit, intracellularly, okay, within the cytoplasm side, intracellularly, who is binding? Sodium is binding intracellularly, and even ATP is binding. And who is binding towards the extracellular side? It's the potassium. Potassium binds to the extracellular side. Okay. Next, what else you should know regarding this sodium potassium ATPase? This sodium potassium ATPase, it's a universal transporter. What does I mean by it's present on all the cells? Present on all the cells. Of course, there are exceptions, but the sodium potassium ATPase are present almost on all the cells. Okay, that's why it's called as a universal transporter. It's present on all the cells. This sodium potassium ATPases, by breaking down the ATP, okay, they can change your basal metabolic rate. Okay, so the sodium potassium ATPases mainly they regulate your basal metabolic rate, means amount of energy required by your body to maintain the homeostasis, means how much energy is getting used by your body in a period of 24 hours in a day. Okay, so just one point, the sodium potassium ATPases, they regulate your basal metabolic rate. Okay, I have discussed this in the endocrine physiology also, how thyroid hormones regulate the basal metabolic rate by using the sodium potassium ATPases. I have discussed this in endocrine also. Okay, now what else you should know regarding the sodium potassium ATPases? The sodium potassium ATPases, they are activated, okay, sodium potassium pump, they are activated by certain hormones and they are inhibited by certain drugs. What are they? First, thyroid hormones. So thyroid hormones activate, okay, thyroid hormones activate sodium potassium ATPases. Actually, the thyroid hormones increases the number of sodium potassium ATPases. They increase number of sodium potassium ATPases. Okay, they increase the number of sodium potassium ATPases, so basal metabolic rate also increases. More ATP is getting broken down, so more basal metabolic rate. And who inhibits the sodium potassium pumps? Drugs called as a digitalis group of drugs. Okay, digitalis group of drugs. Okay, digoxin, digitoxin, okay, cardiac glycosides. They inhibit the sodium potassium ATPases. Digitalis, obein, okay, it's also a drug. Dopamine. Okay, simply remember it like DOD. So these three drugs inhibit sodium potassium pump. So these are some important points which I want you to know regarding sodium potassium pumps as well as primary active transport. Now after primary active transport, now let's discuss about secondary active transport. Okay, secondary active transport. Secondary active transport. So what exactly is this secondary active transport? There is indirect use of energy. Okay, there is indirect use of ATP, indirect use of energy. So why we are calling it as indirect use of energy? I will tell you. Okay, now I will tell you now. See, first of all, let me show you this nephron. Okay, what exactly I am showing here? Here is the nephron. You know, in the nephron, what is the first part of the nephron? Bowman's capsule followed by proximal convoluted tubule. The proximal convoluted tubule continues as loop of Henle, descending limb of loop of Henle. Okay, next hairpin turn will be there. Next, ascending limb of loop of Henle. Okay, now here I am just enlarging the size of the ascending limb of loop of Henle for better understanding. Okay, for better understanding, let me just enlarge this. Okay, here I am just enlarging. Of course, there will be thick ascending part here. This is a thick part of ascending limb of loop of Henle. Now look, this is one epithelial cell which I am showing here. Okay, this is one epithelial cell which is present on ascending limb of loop of Henle. Okay. Now, this membrane which I am pointing here, this membrane which I am just giving an X mark, this membrane is called as a luminal membrane. Okay, this membrane is called as a luminal membrane because it's towards the lumen side. Okay, it's towards the lumen. And this membrane which I am pointing here with the green color, this is called as a basolateral membrane. Okay, basolateral membrane. On this basolateral membrane, look carefully, there is a pump. What this pump is doing is, you should tell me, what this pump it does is, it throws 3 sodium ions out of the cell and brings 2 potassium ions into the cell. Okay, so what is this? This is nothing but sodium potassium pump. So there is a sodium potassium pump on the basolateral membranes of these epithelial cells. So because of this pump, continuously 3 sodium is going out. So what happened to the intracellular levels of sodium? Decreases. So low sodium is present inside the cell. Inside the cell, the sodium levels are less. Okay. So by using energy, see here ATP is involved. This is sodium potassium pump. Here ATP is already involved. By using energy, the sodium levels inside the cell is kept less. The sodium levels are kept less. Okay. Now what happens? Look sir here carefully. Now here sodium is coming into the lumen. Okay. From the proximal convoluted tubule, of course, sodium will be reabsorbed. Almost 70% of the sodium will be reabsorbed. Now the remaining sodium, it is coming like this. Okay, the sodium is coming like this. Now here sodium is there. Okay, when compared to the cell, when compared to the inside of the cell, here sodium is more in the ascending limb of loop of Henle. So sodium is having a gradient. Now sodium can move into the cell. Okay, now here there is a transporter. Now here there is a transporter. Now through this transporter, sodium is moving from high concentration to low concentration. Okay, sodium is moving from high concentration to low concentration. Means sodium is reabsorbed. Okay. Now along with the sodium, not only sodium, even chloride will come into the cell. Okay. Sodium and chloride. Sodium, chloride. And even potassium. Sodium, potassium and 2 chloride. Okay. See, the point which I want you to know is, see here, ATP is involved. See here, the ATP is involved. Now, because of this ATP, we are calling it as active transport. Why we are calling it as an active transport? Because here ATP is involved, but why we are calling it as a secondary active transport? Sir, because of the usage of this ATP, the intracellular sodium levels are kept less. Okay, so sodium is moving from high concentration to low concentration. As the substance is moving from high concentration to low concentration, okay, as the substance is moving from high concentration to low concentration, along with the sodium, even it will drag the potassium and chloride into the cell. Okay. When a substance is moving from high concentration to low concentration, it actually releases energy. Okay. When a substance is moving from high concentration to low concentration, it releases energy. With this drag, the sodium will pull this potassium and chloride. They are actually moving against the concentration gradient. Okay, they are actually moving against the concentration gradient. Okay, who is providing the energy? This is sodium. Okay, the sodium is coming into the cell. Along with the sodium, potassium and chloride are also coming into the cell. Okay, so here any energy is involved. Here there is no energy involved. But where is energy involved? Energy is involved in this area. Okay, energy is involved in this area. So that's why there is indirect use of energy. Okay, for example, see, if you block this transporter, okay, or if the ATP is not utilized, what happens? Intracellular sodium levels will increase. If the intracellular sodium levels are increased, means now this sodium won't come into the cell. If the sodium is not coming into the cell, potassium and chloride will also not coming into the cell. Okay, so for this transport of sodium, potassium and 2-chloride, yes, energy is involved, but in a different site. Okay, so that's why we are calling it as indirect use of energy. Okay, so the sodium potassium 2-chloride symporter, the sodium potassium 2-chloride symporter, which I am showing here, which I am talking right now, the sodium potassium 2-chloride symporter is an example of secondary active transport. The sodium movement, potassium movement and chloride movement, they are all examples of secondary active transport. Okay, so let me write here, wherever you see, how to identify in an easy way, wherever you see the word symporters, okay, symporters are co-transporters. Symporters are co-transporters. Okay, they're all examples of secondary active transport. They're all examples of the secondary active transport. One example is, just now we have seen sodium, potassium, 2-chloride co-transporter. Where is it present? It's present on ascending limb of loop of Henle. Okay. Now, for example, look, sodium glucose transporter type 1 and type 2. Sodium glucose transporter type 1 and type 2. This is also an example of secondary active transport. It's also an example of secondary active transport. Why we are calling it as a secondary active transport? Sodium glucose co-transporter. See, let me give you explanation. See, here, this is the Bowman's capsule. Right now I am showing you the proximal convoluted tubule. Okay. Now, here is the PCT epithelial cell, proximal convoluted tubular epithelial cell. Even in the proximal convoluted tubular epithelial cell, here there is a transporter present. Now, what is this transporter which is present on the basolateral membrane? It throws 3 sodium out of the cell. It brings 2 potassium into the cell by breaking down the ATP. So what is this? This is sodium potassium pump. Okay, this is sodium potassium pump. Now, because of the sodium potassium pump, the intracellular sodium levels are kept less. But if you look the amount of glucose inside the cell, is more. These cells are having more glucose when compared to the outside of the cell. Okay. Now, look what is happening. Here in the Bowman's capsule, sodium is getting filtered. So, sodium is filtered from the glomerular capillaries. Also, the glucose molecules are filtered. Now, look here. So, sodium, here in the lumen, there is more sodium when compared to the cell. Okay, sodium here in the lumen is more. So, sodium is having a gradient. Sodium can move from high concentration to low concentration. So, here there is a transporter through which sodium is moving from high concentration to low concentration. Sodium is moving into the cell. When a substance is moving from high concentration to low concentration, it can take some other substance. So, here now this glucose, which is low inside the lumen, here the glucose is less in concentration. Now, this glucose, it is moving along with the sodium. So, glucose is moving against the concentration gradient. Okay. As the sodium is moving from high concentration to low concentration, it releases energy. By using that energy, this glucose is moving from low concentration to high concentration. Okay. But at the end of the day, see here, where ATP is involved, here ATP is involved, sir. Okay. Because of the sodium potassium pump, the intracellular sodium levels are kept less. As the intracellular sodium levels are kept less, this sodium is coming into the cell. As the sodium is moving from the lumen into the cell, it drags the glucose into the cell. So now tell me, for your exams, the most important question that they will ask you is, what is the name of this transporter here? That there is a transporter which is allowing the movement of glucose and sodium together. What is the name of this transporter? The name of this transporter is called as sodium glucose transporter type 2. On the PCT, sodium glucose transporter type 2 is present. So sodium glucose transporter type 2 is present in proximal convoluted tubule of the nephrons. The sodium glucose transporter type 1 is present in intestines. Okay. So in the intestine, the glucose absorption. You have taken the food will be digested, into glucose. You have to absorb the glucose into the enterocytes. So in your intestines, there is SGLT type 1. SGLT type 2 is present in your nephrons, PCT. So the sodium glucose transporters, sodium glucose co-transport. Sodium glucose transporters are examples of secondary active transport because there is indirect use of energy. ATP is present or ATP is utilized in some other place. Now question is, who is providing the energy? Who is providing the energy for this secondary active transport? It is sodium potassium pump. Yes, true. The sodium potassium pump, they provide the energy for secondary active transport. True. So right. So secondary active transport, there is indirect use of energy, and who is providing that energy? Sodium potassium pumps. Sodium potassium pumps by themselves, they are examples of primary active transport. Sodium is going out, potassium is coming in. So sodium potassium pumps are examples of primary active transport. But sodium potassium pumps provide indirect energy to secondary active transport. Okay. So that's the point which I want you to know. Now a few more important points for your exams. See, this is sodium glucose co-transporter. This is SGLT type 2, right? This is SGLT type 2. Just now I have discussed. There are drugs which can inhibit this SGLT type 2. When you inhibit this SGLT type 2, what happened to this glucose? Glucose will be lost in, the glucose molecules will be lost in urine. Okay. So glucose is going out of the body, leading to glucoseuria. Okay. So what are these drugs which can block the sodium glucose transporter type 2? So there are drugs called as canagliflozin, dapagliflozin, empagliflozins. Okay, so there are these drugs called as dapagliflozin, canagliflozin, empagliflozin. What are they? They are all SGLT type 2 inhibitors. Okay, they are all SGLT type 2 inhibitors. They inhibit the transport of the substances. Okay, they inhibit the transport of substances. Now, what else you should know is these drugs are used in the treatment of which condition? If you block this transporter, the glucose will be going out of the body. Okay, the glucose is going out of the body. So in which condition you want to decrease the glucose levels in the person? In which condition you want to decrease the glucose levels? So these drugs are diabetic drugs. Okay, anti-diabetic drugs. Okay, so they are glucoseurics. Glucose is going out. If you use these drugs unnecessarily, the glucose will be kept into the urinary tract. The glucose is going through the urinary tract. So there will be a chance that this patient who is a diabetic, in whom the immunosuppression will be seen. You know, diabetic people are having immunosuppression, already their immune activity will be less. So there is a chance that they will be getting recurrent infections. So if you use these drugs, there is a possibility that by using this glucose, there can be urinary tract infections. So the side effect of this drug is urinary tract infections. Okay, urinary tract infections can be seen. So these are some important points about the secondary active transport. Now let me discuss about diffusion. Okay, now I am going into the passive transport. Okay, now I am discussing about passive transport. Now passive transport mein the first type is called as diffusion. Diffusion is of two types. You know, the first type of diffusion is simple diffusion. So which substances are getting transported via simple diffusion? First of all, what is simple diffusion? See, usually gases are getting transported in your body. Gases are getting transported. Oxygen is getting transported from your alveoli to the tissues. Carbon dioxide is getting transported from your tissues back to the alveoli. So gases are moving according to their partial pressure. Okay, you can simply say high concentration to low concentration. So gases are moving from high partial pressure to low partial pressure. So that's an example of simple diffusion. So if any substance, if it moves from high concentration to low concentration, it happens by nature. There is no need of any energy. Okay. Example I am taking here. Example, gases. Okay, in your body, that is oxygen transport and carbon dioxide transport. How they move? How they are moving in your body? By use of any energy? There is no need of any energy. Okay, there is no need of any energy for the gases transport. The gases are transported from high partial pressure to low partial pressure. Okay, for example, I am telling you, say in your blood vessel. Okay, which blood vessel? Arterial blood vessel. Okay, in your arterial blood vessel, the partial pressure of oxygen. Okay, the partial pressure of oxygen is 100 mmHg. Okay, it will be somewhere around 97, but I am just taking the round figure. So partial pressure of oxygen is 100 mmHg. So now, what is the partial pressure of oxygen at the level of your tissues? The partial pressure of oxygen at the level of your tissues is, so partial pressure of oxygen in your tissues is 40 mmHg. So by default, by nature, the oxygen will move from arterial blood into the tissues. Okay, so oxygen is getting transported from your arteries into the tissues. So this is an example. This movement of oxygen molecule is an example of simple diffusion. Okay, so gaseous transport is an example of simple diffusion. There is no need of any molecule. No one is involved. Simply gas is moving. That is simple diffusion. Then what is facilitated diffusion? Okay. So in the name itself, it's a diffusion. There is no need of any energy, but this is a facilitated. So let me give you an example for this. Here I am showing your blood vessel. This is your blood vessel. Now after eating food, the food is digested, and what do you have? You are having high levels of glucose in your blood. Okay, you are having high levels of glucose in your blood. But I want you to answer, you require glucose in your blood, or you want the glucose to go into the tissues for the production of ATP? You want the glucose to go enter into the cells. So cells will take this glucose, and the glucose will be undergoing oxidative phosphorylation for the production of ATP. Right? So here this yellow color cell I am showing you. Okay, this yellow color cell. Now by this time you should say, what is this cell, sir? Adipocyte. So here I am writing adipocytes, or you can say skeletal muscles. Okay, adipocytes are skeletal muscles. Now here in these cells, the glucose levels are kept less. Okay, the glucose levels are kept less. So glucose is having a concentration gradient. So this glucose can move, okay, from high concentration to low concentration. But is that happening? No, it's not happening. Because glucose is such a big molecule, it cannot cross the cell membrane. It cannot enter into the cell. Of course, gradient is there, but it cannot go. Okay. So what happens is, look here, whenever you are having insulin in your blood, insulin, what it will do? Insulin is going to act on its receptor. Insulin stimulates its receptor. When insulin is there in your body, when insulin is acting on its receptor, what happens? Now these adipocytes and skeletal muscles, now they will incorporate these channels here. Okay, now they are going to keep this protein channels here. Now what are these things? These are GLUTs. GLUT. Glucose transporter. Okay, glucose transporters, there are different types. On adipocytes and skeletal muscles, there are glucose transporter type 4. Okay, glucose transporter type 4. Now, only through this glucose transporter, this glucose molecule will enter into the cell from high concentration to low concentration. So, glucose is moving from high concentration to low concentration, but this movement, this transport is facilitated by this protein, which is called as a glucose transporter type 4, as the transport is being facilitated by a protein. Okay, by a protein molecule. It is called as facilitated diffusion. So, who is facilitating this diffusion process? Movement of substance from high concentration to low concentration? A protein molecule, maybe a channel, maybe a carrier protein or something. It's helping in the transport of the substance. So, that's why it is called as facilitated diffusion. So, very important point for your exams. There is a lot of chance that not even I should say 100% chance, definitely this question will be there in your exam. Either SGLT or GLUTs. One question will be there, sir. Okay. So, all your glucose transporter 1, 2, 3, 4, 5, 6, 7, something like that. Glucose transporters, all glucose transporters are examples of facilitated diffusion. Okay, how to remember? Just remember GF. Okay, you know what is GF? Girlfriend kind of thing. So, glucose transporters are examples of facilitated diffusion. Okay, symporters are examples of, symporters are co-transporters. Symporters are co-transporters are examples of secondary active transport, S for S. So, symporter for secondary active transport. GF, glucose transporters are facilitated diffusion. Okay, so facilitated diffusion is also completed. Now, what we are left with? Osmosis. Okay, osmosis. Now, let me write here. So, what exactly is osmosis? Which substances are getting transported? See, which substances are moving here? Water. Water movement, not gases, water moves along the osmolarity, according to the osmolarity. Okay. For easy understanding, now look here. Here I am taking a beaker. Okay, here I am taking a beaker. Now, here I am keeping pure water. Imagine I am keeping here pure water. So, what is the osmolarity? Does it have any concentration, any osmolarity? Osmolarity means how many ions are present in a particular amount of fluid, in a particular, for example, how many ions are present in 1 liter. Okay, osmolarity, sir. Now, here there are no ions, it's a pure water. So, the osmolarity is 0 milliosmoles. Okay, 0 milliosmoles. Now, it's pure water molecules. All the water molecules are free. Okay. Now, when I add these electrolytes like sodium, chloride. So, if I am adding this salt, sodium, what happens? Now the water molecules will be occupied. Now the sodium is going to bind with the water molecules. Now the number of free water molecules are decreasing. Okay, when I am adding this sodium, what happened to the concentration? First thing, what happened to the osmolarity? So if I add sodium or electrolytes, osmolarity increases. Okay, what I am adding? Sodium, these are solutes. Now I am adding solutes into the solvent, into the water. I am adding the solutes. Okay. So this osmolarity is because of the number of solutes. More the number of solutes, more osmolarity. Okay. Now look, if I am adding the solutes are binding with the water molecules. So the number of free water molecules decreases. Number of free water molecules decreases. So I can say something like this: more the osmolarity, okay, if the more osmolarity means there is less free water molecules. Okay, less number of free water molecules. Okay, now based on this concept, you should tell me, okay, you should answer this. Here I am taking a beaker A. Okay, here I am taking a beaker A. Here I am taking this beaker B. Two beakers I am taking. Okay, beaker A and beaker B. Now I am just filling them with water. Okay, with different osmolarities. Beaker number A is having an osmolarity of 100 milliosmoles per liter. Beaker number B have 10 milliosmoles per liter. Okay, two different osmolarities. Now I am just connecting them. Okay, now I am connecting these two beakers. Now tell me, water will move from A to B or B to A. Now most of the students will think, so usually substances will move from high concentration to low concentration. No, here I am talking about the water. You say more osmolarity means here more sodium molecules are there. More sodium molecules are there means less free water, less water molecules, less water is free. Okay, less number of water molecules are free. Here 10 milliosmoles means this is less concentrated. Beaker number B is less concentrated, which means here more free water molecules. Okay, more number of free water molecules. So in beaker number B, there are more free water molecules available. In beaker number A, because of the more osmolarity, there are less number of free water molecules available. So that's why here the water will move from B to A. Okay, so osmosis is movement of water. Okay, let me write. So osmosis is movement of water from low osmolarity to high osmolarity. Okay, movement of water from low osmolarity to high osmolarity. Okay, you can understand it something like this in an easy way. Here I am telling you beaker number A have high osmolarity 100 milliosmoles, which means more sodium is there. And you know from your basics, wherever there is sodium, water moves towards the sodium. Okay, here there is less sodium, here there is more sodium. So water always moves towards the sodium. The solutes drag the water. Okay, so again I am telling you osmolarity is dependent on solutes. More the number of solutes, more osmolarity. Less the number of solutes, less osmolarity. Okay, so water will move from low osmolarity to high osmolarity. Don't think like water will move from high osmolarity to low osmolarity. No, water will move from low osmolarity to high osmolarity. Low osmolarity means more free water. High osmolarity means less free water molecules. Okay, so water moves via osmosis. Water moves via osmosis. Gases moves via simple diffusion. Okay, done. Now after this, let me discuss a special type of transport which is called as vesicular transport. Okay, vesicular transport. Same, vesicular transport means here the substances are transported across the cell membrane in the form of vesicles. Which substances? Large substances. Large substances are transported across the cell membrane okay, large substances are transported across the cell membrane in the form of vesicles. Okay, in the form of vesicles, right? Same, there are two types of vesicular transport. Vesicular transport is of two types. One is exocytosis and endocytosis. Okay, exocytosis and endocytosis. Exocytosis means the substance is the large substance, for example, a protein or a neurotransmitter or something. A substance is transported in a vesicle out of the cell. Like a substance is moving out of the cell in the form which is packed inside a vesicle. Maybe it's a hormone. Okay, so exocytosis means movement of substance out of the cell. Okay, endocytosis means you are taking a substance into the cell in the form of vesicle. Okay, so endocytosis means movement of substance into the cell. Okay, so exocytosis, endocytosis, one is sending out of the cell, one is taking into the cell. This exocytosis, okay, what do you require for your exam? Exocytosis process के लिए you require certain proteins for the formation of this vesicle, and for the process of exocytosis you require certain proteins in the cell membrane which are called as snare proteins. So which proteins? Okay, snare proteins like synaptobremin. Okay, snare proteins are required for the purpose of exocytosis. And one more question which was asked in the exam is exocytosis process के लिए, for this exocytosis process, which ions are required? Calcium is required for exocytosis. Okay, snare proteins, exocytosis, calcium, exocytosis. And endocytosis. See, endocytosis, it's of different types. One is phagocytosis, pinocytosis, receptor mediated endocytosis, okay, these are different types of endocytosis. Now, what exactly is phagocytosis? For example, say here is a neutrophil, or for example, say let's say there is a macrophage. Okay, here is a macrophage and here is a bacteria present. Okay, here is a bacteria present. Now, this macrophage, what it can do? It can go and do the engulfing of this bacteria. Okay, so what this macrophage will do is, this macrophage, let me show you here, it's going to extend its cell membrane. It's going to extend its cell membrane, okay, around this bacteria. It's going to extend its cell membrane around this bacteria, and this cell membrane, it's going to form a vesicle. It's going to completely enclose the bacteria, and this bacteria will be taken into the cell. Okay, finally what happens is this macrophage is going to take that bacteria into the cell, okay, by forming a vesicle. So now it is into the cell. Okay, so this vesicle, now it is called as a phagosome. Okay, so what this macrophage have done? So macrophage, it got hungry and it have taken the bacteria. So this is called as a cell eating. Phagocytosis means cell eating. Okay, cell eating. Who does that? Neutrophils, macrophages, these phagocytic cells. And after eating, we'll have the feeling of thirst. So pinocytosis is cell drinking. Okay, so what the cells will do is, the cells are going to be covered with this extracellular fluid. There is extracellular fluid around the cell. So what the cells will do is, sometimes when the cells are getting dehydrated, now the cell again is going to extend its membrane, okay, into the extracellular fluid, and the water will be taken in. Now this is cell. Now what the cells are doing? The cells have taken fluid into the cell. Again in a vesicle, the fluid is coming in. So this is called as penocytosis. Penocytosis. Okay. If I am saying reverse penocytosis, cell is releasing the fluids out of the cell. Okay. Penocytosis, it's a type of exocytosis. Okay. But anyway, so phagocytosis, penocytosis, they are a type of endocytosis. The substances are taking in, bacteria is coming in, the fluids are coming into the cell. Okay. And what is this? Receptor mediated endocytosis. Say for example, now here is one cell. Okay, here is one cell. Now here are the receptors present. Okay, here are 4 receptors present. For example, now imagine there is a condition called as pheochromocytoma. Pheochromocytoma. In this condition called as a pheochromocytoma, there is excessive amount of neurotransmitters, excessive amount of epinephrine is there. Okay, excessive amount of epinephrine or adrenaline is there. Now this adrenaline is going to act on these receptors. Too much stimulation, too much. Okay, the neurotransmitter is stimulating this receptor, stimulating this receptors. Too much stimulation is not good. So what these cells will do is, cells are going to form a blob like this. Okay, so what the cells are doing? The cells are forming a blob. So now what happens? Look carefully. Okay. So now what happened to the cell membrane? The cell membrane is invaginated. Now what are these two? These two are the receptors. Now here are the 2 receptors. Ultimately what happens is, now this cell is decreasing the number of receptors. Now that membrane was totally invaginated, and ultimately both these ends will fuse, and now here you are having a vesicle with these receptors. So what happened to the number of receptors on the cell surface? Decrease. From 4 it become 2. Okay, initially how many receptors are there? Initially 1, 2, 3, 4 receptors are there. Now there are only 2 receptors. So whenever there is excessive stimulation, the number of receptors on the cell surface can be decreased. So with a process called as endocytosis. This is called as a receptor mediated endocytosis. Okay, so this is a type of endocytosis. Penocytosis is a type of endocytosis. Phagocytosis is a type of endocytosis. Endocytosis process के लिए you require certain proteins. Exocytosis require snare proteins. Endocytosis they require proteins called as clathrins. Okay, clathrins. So clathrins and caveolins. Clathrins and caveolins, these are the proteins which are required for the process of endocytosis. Okay, and obviously vesicular transport is not a passive transport. Definitely here also ATP is required. Okay, here also energy is required. There is no doubt in that. Vesicular transport also requires energy. So with this we have completed different types of transport. Again I am summing up important points which you need to know. Active transport, 2 types: primary active, secondary active. Primary active, how to identify? If you see the word ATPases or pumps, ATPases or pumps, they are all examples of primary active transport. Secondary active transport, how to identify? All co-transporters, sympoters. Okay, all co-transporters: sodium, potassium, 2-chloride sympporter, sodium chloride sympporter, sodium glucose co-transporter. All they are examples of secondary active transport. Facilitated diffusion, examples: glucose transporters, all glucose transporters, GLUT1, GLUT2, GLUT3, GLUT4, GLUT5, all are examples of facilitated diffusion. Gaseous transport is simple diffusion. Snare proteins are involved in exocytosis. Snare sounds like snake. Okay, snare sounds like snake. Okay, so who is the snake, sir? Okay, who is the snake? Your X. Your X is the snake. Okay, that's why she is X, right? So remember, snare proteins, exocytosis. Exocytosis require calcium, very much important. Okay, and clathrins are required for endocytosis. So with this, the topic of transport across the cell membrane is completed. Hope the video is helpful. Thank you.